Good afternoon, and thank you all for standing by. For the duration of today's conference, all participants' lines are on a listen-only mode until the question and answer session. At that time, if you would like to ask a question, press star 1. Today's call is being recorded. If you have any objections, you may disconnect at this time. It is my pleasure to introduce Ms. Irene Ihir. Thank you, ma'am. You may begin.
Thank you. Hello, I'm Irene Ihir of CDRH's Office of Communication and Education. Welcome to the FDA's 31st in a series of virtual town hall meetings to help answer technical questions about the development and validation of tests for SARS-CoV-2 during the public health emergency. Today, Timothy Stenzel, Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality, and Toby Lowe, Associate Director of the Office of In Vitro Diagnostics and Radiological Health, both from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to today's discussion. Please remember that we are not able to respond to questions about specific submissions that might be under review. Now, I give you Timothy.
Welcome, everyone. A pleasure to be on the call again with all of you. So appreciate all your creative minds and dedication to addressing unmet COVID pandemic needs. We look forward to working with you. You may start to realize that you have additional folks who you've not met before. We've been hiring and we've been shifting some resources to try to work through all of the volumes of submissions as quickly as possible. We've been doing that since day one, but we continue to do that. So hopefully that has a net positive influence on all of this. Just to start off, I wanted to talk about something that's very important to all of us as test developers and those involved with validation and authorization and use of these tests in this pandemic. It also applies to non-pandemic situations. I do want to help enlist your support and help in educating all of the various stakeholders who utilize these tests in different situations. So I want to talk about false positives again. We continue to hear about challenges relating to getting false positive results, and I would add even in situations where it's totally expected. That might seem odd. So first of all, I'm not aware of any perfect test. And I want to go back to my example of using a test, whether it be an antigen test or a molecular test. Neither, I don't know a single one that's perfect, that is 100% specific. That is, every time a patient is truly negative, the test is negative. It just doesn't exist to my knowledge. So if we just assume for a second, just for the math, that that the specificity of any of these tests is 99%. That is, 99 times out of 100, a patient who is negative will test negative, and only 1 time out of 100 will a patient who is negative test positive. I think that's a very high specificity for almost any category of test. The challenge, of course, is if we begin to test in large-scale screening, whether it's antigen or molecular, and we're screening populations that, in particular, have a low percent positive rate. If that percent positive rate falls below the 1% false positive rate, it means that even for a perfectly well-functioning test, it's a well-designed test, it's a well-validated test, and the users who are performing the test are following the instructions. This according to the package insert, they are going to get a 1% false positive rate if the test is 99% specific. And it really doesn't matter if the patient is symptomatic or not. If they're asymptomatic, it applies the same. And it doesn't even really matter if a test has achieved a claim for screening asymptomatics. Specificity is specificity. It's typically going to be the same in all these different populations for a given test format, a given sample type. And so the challenge is what do you do when the false positives, which are totally expected, outnumber the true positives? I think it's important to say this is a totally expected situation, and we plan for it. We plan for it by, in those situations where the prevalence of positive disease, where the percent positive is low, significantly less, or even even if it's a 1% positive rate, you're going to half your positives are going to be false positives, half your positives are going to be true positives. So unless your percent positive rate is pretty high, the idea that you might have a false positive, if it's going to impact somebody's livelihood, that is, they may be asked to quarantine themselves for 14 plus days, or if it involves putting a patient into a risky situation, or healthcare workers aren't alerted to a patient having this, or it determines outcome. If they have flu, they're going to treat somebody this way. If they have SARS, they're going to treat them another. So where all those situations matter, and your population is such that the false positive rate is not insignificant, then it's important, and I would recommend that you get some sort of orthogonal confirmatory test done. I would prefer it be a molecular test of high specificity and sensitivity. There is the option potentially of using another direct antigen test if your primary test is a direct antigen test. What I don't think we have a good handle on yet is whatever causes a false positive for one test, particularly a test that doesn't have an extraction of nucleic acid step up front. If there's something in that sample that lends itself to a false positive result in a given individual, if you just solely go to another different orthogonal direct antigen test, I just don't know at the moment, and maybe somebody does. If you do, please reach out to us through our template email address. If that second direct antigen test will give you a true result based on the sample having certain potential characteristics that may lend itself in a direct antigen test to giving you a false positive. So I don't know. I think I personally lack the information to give a recommendation on that. Be very interested in seeing data about two orthogonal direct antigen tests, and when you have a false positive, can you use that orthogonal test to determine that. The other thing is, if there is any sort of loss of sensitivity with a direct antigen second test, a highly sensitive, highly specific molecular test is more likely to give you an accurate confirmation of whether you have disease or not. So that's the introductory remarks for today. Thanks again for joining us, and I look forward to your questions. Thank you so much.
Operator will now take questions.
Thank you. If you would like to ask a question, please make sure your phone is not muted. Please press star one, and when prompted, clearly record your first and last name so I may introduce you. Again, to ask a question, press star one. Our first question is from Alberto Gandini. Sir, you may go ahead.
Hello. Hi. Can you hear me?
Yes.
Yeah. So my question is related to the what's the step the NIH does to ensure, the FDA does to ensure that the validation done by the NIH are done according to the instruction of the packages of the assay that is being tested. We know in at least in one case, the NIH did not follow instruction as far as, for example, using sample matrices that are not intended to be used in the assay. For example, using different plasma coagulated with different anticoagulants as the one that the sponsor was actually providing and validated. It seems to be, to me, a pretty serious violation of the instruction of use. And also, we now follow, in some cases at least, again, at least in one case we now use the proper specific control to ensure that the operator knows how to use these tests. Some of these tests may be new technology, and so the operator may have to sort of train on it. And I'm wondering, those can have serious impact because you can end up to basically, validating correctly certain, either in a good way, in a bad way, whatever you like to see it, the essay that has been presented. So I would like to know what kind of steps the NIH does, and if there is any concern about the NIH mishandling of the assay during the validation, if there's any possible way for the sponsor to discuss that either with the NIH or with the FDA or all three together. Thank you.
First of all, the NCI is doing a fantastic job and such an important job. We have processes and procedures in place to try to ensure that in all cases package inserts are followed and that the correct sample types are used. We don't have the luxury of getting a wide variety of panels for each different kind of sample type as far as serum or plasma, and then as far as for plasma for the anticoagulant that's used. And so we are somewhat beholden to the panel as it gets formulated, and therefore we do want to look to those situations where a non-ideal sample type was used. We typically can look to aid us, look at a subset of the results that match and line up with the IFU for the product. If that is missed, when we get the results, we do reach out to sponsors with their NCI results, and that is an opportunity for sponsors to engage in a conversation with the FDA to address those specific questions. And a few times it has come to my attention of this, and we've done our best to address those concerns. So that's an opportunity for sponsors when we give you that feedback on the results. The panel does use serum and plasma, and it does for plasma. It does use the common anticoagulants in general. So and there may be some tests that have limitations on serum versus plasma and vice versa, and for plasma the type of anticoagulation. So we appreciate working with sponsors to help us if we've overlooked something, either at the NCI or at the FDA, and we'll do our best to properly address that situation. So if you feel that's not the case, you can always reach out to Toby and me through the template's email address for a specific discussion and a specific situation.
Thank you.
And our next question is from Lewis Perlmutter. Sir, you may go ahead.
Yes, thank you, Ted and Toba. The question really concerns whether there are any approvals for recently submitted rapid antigen immunoassay tests for the SARS-CoV-2. I'm not thinking of the Biomex Now test by Abbott, but any others. And are there any in the works?
So we have posted all of those that we've authorized on the IVD EUA website at the FDA. I believe there's 6, and I believe Quidel came in subsequent to the Binax Now test with a flu AB SARS test. So you can always see what's within, we post that within, an hour to 24 hours, or, in business days after authorization. It happens over the weekend, and it usually happens it may happen not until on Monday. And there are a number of developers working on additional rapid antigen tests. And I can't go into any more specifics than that, but
yeah, because I was aware of those. That there are a few submissions. Yep. Okay. Thanks very much.
Mm-hmm.
And our next question is from Alexis Sauer-Budge. You may go ahead.
Hi, this is Alexis Sauer-Budge from Exponent. I was hoping that you could give us a little bit more detail with regard to the announcement about the FDA declining to review LDTs. You've also stated that it's a high priority to review high-throughput tests. So if a CLIA-certified lab is developing an ultra-high throughput molecular test, would you still review the submission?
So we are currently declining to review LDTs, and for the reasons that we stated on our FAQ page, and that is largely because they the FDA cannot require an EUA, and according to the HHS statement. As far as high throughput goes, those are for those tests that then we choose to review due to the unprecedented number of applications. We want to focus our resources on those that do require an EUA, and so a single lab that's developed their own test and happens to be high throughput, doesn't, if that's, sort of the specifications of that, it's our understanding it doesn't require an EUA. And so we are focusing on high throughput situations now. Some of that's, going to be, kits that have incredible amounts of automation where you load samples and step away, and they have a high throughput. So when we talk about high throughput, being a high priority, it's that sort of situation where, in particular, distributed kits. So if the kit is high throughput, this instrument is high throughput, and it's able to be offered in a lot of different labs, that's going to have a huge positive public health benefit, and that's where our attention is going to be focused among the other high priority items. And those items that do require an EUA prior to launch. Toby, do you have any other comments?
No, I think exactly what you said. We're focused on distributed tests, and that includes high throughput distributed test kits.
Thank you for that. If I could just have a follow-up then. If the intent, though, was to provide the protocol to other laboratories, say, so, buy these particular pieces of equipment and use this protocol sort of similar to the Yale Saliva Direct protocol, is that an avenue that would make it not an LDT and the FDA would review it?
So we can't, without all of the information that for a specific scenario, it's difficult to give an absolute answer. But I can tell you that the Saliva Direct test that you're referring to is considered to be a distributed test.
And not an LDT. In fact, I don't believe that AEL actually has a lab that the developers don't have a lab that they're doing clinical testing in. So they are basically a kit that has the instructions, and then everything needed for the kit is required but not provided, and that's the way we view that.
Great, thank you so much, and thank you for all the hard work that you're doing to help us with this pandemic.
No, you're most
Thank
welcome.
you.
Thanks for those kind words.
And our next question is from Christopher Benson. Sir, you may go ahead.
Hi, Tim. I'm calling again to update you since we last spoke about this about 2 weeks ago, about shipping the SARS-CoV-2 FDA panel overseas. We've made substantial progress. I've worked with Dr. Myra Garcia at your office. But we did receive a request from the foreign country for a Certificate of Inactivation from FDA for the panel. I thought this was a reasonable request. Do you know if you have that kind of certificate?
Toby may know. You know, we have processes and procedures within the FDA. We received live virus. We grew it up on the FDA campus in BSL-3 facility. We followed a protocol, an institutional protocol that was reviewed by the FDA to make sure that it was safe for our employees. And I think we did the same for the inactivation. What I don't know is, and so we have complete confidence that it is safe to distribute as a BSL-2 level panel. But what I don't know is if we have the ability to provide such a certificate. And there may be an opportunity to write a letter rather than, say, provide a certificate. So, and I'm not sure if you've had that conversation with Myra. It's not coming to mind. I meet with Myra a couple times a week, if not more frequently. Toby, you know anything more? And
I don't know that whether or not we have that certificate, but we can. Myra would definitely know. She's just not online at the moment because she's in the lab, I believe. But we can definitely find that out.
All right, thank you. Yeah, I wrote to Myra, and so maybe a letter would be sufficient. The other thing they recommended is a limit of 10 kilos for the dry ice shipment, which I'm sure is due to international flights. But I'll follow up with Myra on that. Thank you, Tim and Toby.
Well, that's not a lot of dry ice, especially if you're shipping international, which I have, fortunately or unfortunately, substantial amounts of experience with, having been a chief operating officer that has shipped internationally in my previous role. So the other thing is that we have worked with some sponsors who have found a facility or lab that they can work with in the U.S. To do the testing for them, and where there were challenges in shipping internationally. So that is a potential avenue as well. It's not required or even recommended. It's just a statement of fact.
Okay, I appreciate that.
Thank you.
Thank you.
And our next question is from Elliot Milleton. You may go ahead, sir.
I had a couple of questions. One, is there a panel for antigen test validation or a rapid antigen test?
Not yet to my knowledge. And there have been fairly detailed discussions about what we can do in this area. One of the concerns relates to the prior conversation about the fact that this is a BSL-3 level virus, and most folks don't have a BSL. Most developers and labs don't have a BSL-3 facility so that they can safely handle it. There are some, but most don't. And therefore, I'm just going to tell you the transparent issues that we're facing. So you can go ahead and inactivate that virus in a number of different ways. And then you want to make sure it actually still functions at the antigen level. And it's going to be challenging to provide that kind of assurance that it's going to behave just like wild-type live virus. But we are looking into it, methods of inactivation to make it safe. We are looking into how well it performs. We're also looking at potentially the opportunity to express antigen and make protein. And then obviously you get away from the need for inactivation because it's not whole virus and should be very non-toxic. So we're working on it, and I'll say that we're working on it within the U.S. government, and there may be others outside the government who are working on it. We're just not ready to move forward, but we certainly are very interested. So thank you for your question.
Thank you. And when you say you'd have to make sure the antigen still functions, first of all, do you have any preference for heat inactivated versus radiation inactivated, and how would you recommend determining whether it's still functioning?
Yeah, so I don't know that I have a preference there as far as I think the way to go about this is to try potentially different methods, different conditions, and see if it see how robust that is, and then see how it impacts at least the EUA authorized antigen tests. But again, they're not cookie cutter. The antidirect antigen tests are not cookie cutter, and if they're using antibodies that are different, those antibodies then in most cases are going to see a different epitope, and that could be different epitopes could be variably impacted by different methods of inactivation. Likewise, if you begin expressing proteins, if it doesn't, if the proteins aren't processed and get the non-nucleic acid directed modifications in the same way as in human cells, then there could be alterations in performance. So they're just very clearly additional challenge when you try to develop a direct antigen reference panel as compared to a reference panel for molecular that we can basically know that we inactivate, and know that if the RNA is there and stable, that we will have uniform good results.
Thank you. And one more question for validating an antigen test with saliva. Can it be compared to a PCR test with saliva? And do you have a preference as to whether it's a saliva sample for PCR or a nasal swab sample?
I think that's a great question. I think it's important to, whenever you're doing these kind of comparative tests, to just make sure with the FDA staff in our office that it's an okay comparator. I would say that if it's an EUA authorized high sensitivity molecular method, there's good chances that you could use saliva as the comparator, molecular saliva, for the antigen comparison. But I would say it can depend, and just double check with our team.
I don't have it up in front of me right now, but I believe the antigen template discusses appropriate comparators and specifically mentions no saliva. So if you are particularly interested in using saliva as a comparator, I would recommend reaching out to the antigen team prior to beginning that study to discuss that.
Great, thank you.
That would be an alteration to what's recommended. And, over time, our thinking can change, and we try to remain open. Now that we have a lot more saliva experience, the team's thoughts may have changed. I'm not going to speak for them, though.
Thank you very much.
Our next question is from Edward Strong. Go ahead, sir. You may go ahead.
Hi there. Thank you. Yes, Edward Strong from IASA in San Diego. So we have a handheld portable reader instrument that reads lateral flow devices and other formats of rapid tests. IASA's models, we make that instrument and then we work with partner developers of those testing devices. I think you probably remember on last week's call, you indicated that there may soon be some guidance on instruments, especially now that there's recognition that instruments like a reader would be beneficial to improving or preserving the sensitivity and reporting in off-site testing. I was just wondering if there was any update that you might have to provide on that guidance on that front.
Yeah, so Toby wasn't here last week. Terribly missed her. And she may know the latest. It was my desire to have some additional recommendations. 2 weeks ago, Toby was here. I wasn't here. I believe that we made some adjustments to the template, the appropriate templates, but those have not been posted yet. And I don't think we can say when it can be posted, but I do believe that guidance can be given, and this would, if I remember correctly, this was more for serology tests. But potentially the same could be applied to direct antigen tests. But our thinking was around, and my input was around what to do for serology tests. So I believe an email to the FDA staff can provide some recommendations based on our current thinking until that guidance is posted. We're obviously. Toby, you have anything else to add?
Yeah, I think we've had some emails with you as well on this topic. I think that the main point is going to be that we need to see evidence that the reader works with a specific test, and so that's what we're going to be most interested in. We do have some experience with readers outside of the public health emergency where the readers have had potentially can have a negative impact on a test performance. So we do want to see those paired together. And we would be happy to work with you on the best pathway to do that, whether it's working directly with the EUA holder for the test, where they could come in with an amendment to or supplement rather to their EUA. We can, we're still having some discussions internally about different options there, but that's something that we can work with you on. But the biggest point is that we really do need to see the data with the test and the reader paired together.
Okay, yeah, if I could ask a follow-up question, because that's particularly the area that we were thinking about. Obviously we've been working with some developers with their EUA submission, but yeah, we were looking at IASA for guidance on if we could supply data that would preserve the performance of these tests, but do it independently of the developers, because our model is very much like an independent open platform for our reader to be functional in many different serology or antigen tests. So it sounds like you're having those discussions internally, and maybe we'll reach out again and get some guidance on that direction.
Yeah, and so Toby was talking about accuracy determination, and I was talking more. That's important as well, but I was thinking a lot more towards the flexibility and regulatory pathways for some for reader developers, and what might be required of the reader developer that could be independent, or recommended for reader developers that could be independent from the lateral flow test itself. In the end, just to be transparent and clear, it would, unless you were to repeat all of the validation studies that the original manufacturer did for their EUA authorization, get your own EUA authorization, you're going to want to rely on the data that's already been accumulated and the EUA authorization if it's authorized for that test. And that will require some sort of letter from the test developer to the FDA saying that they give permission for a particular instrument developer or reader developer to use that data in their submission to have a reader that can specify that it works with that particular lateral flow. So that was a lot of words, but basically in the end, even if you do things independently, and I have suggested edits that would allow some independence so that it makes it as easy as possible to add readers, it still requires collaboration on some level with the company that developed the lateral flow device.
Of course, this is perfect, very helpful, and I echo what everybody else has said. Thank you guys for the sleepless nights, and you all deserve vacation. Thank you.
Thanks so much.
Thanks.
And our next question is from Kodamudi Venkep. Go ahead, sir. Your line is open.
Hi, good afternoon. Can you hear me?
Yes.
Yeah. Thank you for taking the question, and thank you for all the updates. My question is related to the initial remarks that you mentioned about the false positives. If you are giving an example of 99% specificity, 1 out of 10 is a false positive. And at the end, you were mentioning about if antigen test is there, you can do orthogonal test with the secondary test. Is it only for the antigen test or even for molecular test? So any positive test, if there is it can be potentially false positive that needed to be confirmed with another test. I did not get what you were mentioning clearly. Can you please elaborate and clarify that doubt, please?
No. This also applies to molecular tests. They're not perfect either. Developers do their best. Users of those tests do their best. In my conversations with those who are doing pure surveillance, which for this pandemic is not something under the purview of the FDA, but yet we still have a lot of conversations. This is typically done on a lot of different educational campuses around the U.S. They oftentimes use molecular means, and the ones that I've spoken to specifically about this, they do have false positives, and they do. First of all, if they have a positive, they want to move it into a CLIA lab to be able to, in some situations at least, to provide a CLIA result rather than an unregulated surveillance result, which is cautioned against, obviously. But they have found that they do want to do that confirmatory testing. So, in particularly, as I said, in a low % positive population. And I have talked to some who have rates of detection of positives that are well below 0.1%. So when you start to get into a positivity rate, well below not even 1%, but well below 0.1%, you are absolutely going to get into the territory with even extremely high specificity tests. You're going to have some false positives. So you do want to do some sort of confirmation. Hopefully that's helpful.
So it is only, when if it is you are 99% specificity, so only if you are testing a population where it is less than 1%, then this approach is useful. It is not for other diagnostic tests, even if it is.
I think it's something that every clinician, every healthcare worker that interprets positive and negative results of testing, no matter what kind of testing it is, needs to keep in mind. It's a very important thing to keep in mind. It's one result, and it could be a false positive, it could be a false negative. What we do at the FDA is make sure that these tests perform at a certain level, and we make public that performance information so that the users of these tests, whether it be labs or it be clinicians, can make an informed decision about what does a positive result mean in this patient, in the population that I'm testing. Is the patient symptomatic or are they asymptomatic? Have they been exposed or not exposed? So this is a very complex science to properly interpret the results of tests. What I was really urging folks to do is not to assume in low incident populations that a positive is a true positive, just due to the chance of false positives. So as your percent positive rate increases, say it increases, there's some states I'm saying, wow, they're over 30% now, right? Let's just say 20%. Let's say percent positive in the population is 20%, and your false positive rate is 1%. It means that you're going to have 21% positive rate. 1% of that is a false positive. The rest of it is true positive. So it means that, or it was just saying overall the measured rate is 20 to make the math easier. So that 19% true positive rate, 1% false positive rate, your percent positive overall, including the false positives, is 20%. It means 1 out of 20 is a false positive, or 5% is a false positive. But you just, if you're a clinician interpreting results or your laboratory interpreting results, you just want to know that. What is the chances of this individual result based on the performance of the test I'm using that it's a false positive versus a true positive? And so you never get rid of the false positive issue because no test is perfect. You never get rid of the false negative problem, no matter what the incidence is, because no test is perfect. So I'm sorry I can't give you a really cookie cutter, applies in all situations answer. It's just important that if it really matters for the result, it might be good to get a confirmatory test.
So just like how you recommended the serology test, you have certain disclaimers. So do you mean these molecular test results also have to come up with these situations or the disclaimers?
We are looking at, Toby's working on an FAQ update as we speak. In fact, I think she sent me language to review, and I haven't done it yet. She sent me language yesterday. I forget when. So we're going to try to further explain this, and we may look at additional information in the intended use statement, especially when, say, asymptomatic claims are made about a test. So it really is situational. It depends. It depends. So what we saw is that recently is nursing homes were seeing a lot of positives. When they went and tried to confirm it, roughly they were only able to confirm about half of those positives, and they were shocked. They were frightened that that the test wasn't performing. What I'm saying is the test performed, to my knowledge, in those situations, the situation that I'm aware of, I don't know that the test, I believe the test was performing as intended. It's just that this math says that when you have a low percent population, you're going to have false positives. So don't expect false positives in that situation. And therefore, if it's really important to know whether that patient's positive or negative, get a confirmatory test.
Thank you very much for your explanation. I appreciate it.
And you're welcome. And the reason I enlist this audience of developers is that it can negatively impact what we do if assumptions are made based on performance that's entirely expected, that there's actually a problem. There isn't a problem with the test. And so if someone views there's a problem with a specific test or a class of tests, like direct antigen tests, when there isn't, just because they got false positive, then it has potential for the public to not be supportive of the testing schemes that we want to roll out in order to help patients, especially those who are most vulnerable. We all read stories of how SARS can go around a nursing home in no time flat, and everybody's infected, including some of the staff. And that's what we're trying to prevent with some of the schemes that we're using, and not just us, but others. All right.
Thank you.
Next question, please.
And our next question is from Dana Hummel. You may go ahead.
Hi, my question is regarding the sale of COVID diagnostic tests to researchers for research use only. And I wanted to ask specifically if there's any restriction by the FDA to sell a non-EUA kit to a research lab for research use only. I understand the FDA recommends the use of EUA kits by researchers, but I'm wondering if researchers are allowed to purchase non-EUA kits for their research.
Toby, do you want to handle that question?
Yeah, sure. So if it is truly a research lab doing research, then absolutely they can use research kits, research tests, research components. What we don't want to see is kits that are labeled for RUO being sold to a clinical laboratory for clinical use. And we have discussed previously the guidance document that we have that talks about appropriate labeling for RUO, and that is a really good resource to take a look at how we've laid out what is appropriate for an RUO-labeled device versus a device that should be IVD labeled.
Perfect. Thank you so much.
Sure.
We will take our next question.
And our next question is from Winley Ju. Your line is open.
Hello, thank you for taking a call. So I just have a question, continue the discussion on the molecular test, false positive. So right now, I think it's very tricky here to determine the false positive because we don't really have a good reference panel. And what we do is just compare two molecular tests with each other. So I could say one's false positive or another one's just a false negative, you know. It's always hard to see which one's true, false positive or false negative. So what I found is that with a higher CT, this could be a problem with a very high CT value. And so with that being said, like false negative, can I see that false positive could just have more, could happen at the higher CT more, and then just lower CT?
Yeah, I think that may be dependent on the test and the platform, and in particular the detection chemistry, because if you have a baseline that say is set such to sort of maximize sensitivity, that baseline can be inadvertently in a small, in a well-performing test, a small percentage of cases, that the baseline can be crossed earlier on too. and just in case based on the detection technology. So really, my statement has to do with when you know you have a population that has a low % positive, and it's critical to properly to know whether that patient is truly positive or not. And when the chance of a false positive is significant, even with the test performing as intended, that's when I recommend that you take a look at confirming. This is not confirming all false negatives, potential false negatives, and all potential false positives. That would be not workable. But when you are interpreting results of any test, the clinician wants to take that into the context of the situation. So if they think this test is falsely negative, I know that some of them are ordering a second test. Or if they think it's falsely positive, I know that some of them are checking to make sure. So this at least for some percentage of the clinical population of healthcare providers, they are taking this into account. I just think that due to recent news reports and other reports, there are elements of the healthcare system that we're trying to bring up to speed on, in particular, interpreting positive results in a very low % positive environment.
Yes. Yeah. So if you have a very low positive rate or prevalence, then you can afford to check every positive. But if you have higher prevalence, and also if the CT value are high in many people, and then it's kind of hard to do that. And also if you don't check them right away, and then if you check like a few days later, and that could just be that first time is real, the second time is it's still real, but the virus just clear out,
You're exactly right. You're exactly right. It's why basically every test that we've authorized to date is by prescription. Mm-hmm. Why is that? Because having a clinician involved, somebody who can order the test involved, they can help interpret the results correctly. Is it a perfect system? No. But even the direct antigen tests that are being used in nursing homes across the country are still by prescription only. And so we are wanting to augment what we're saying. The CDC has guidelines on this that are published on their website, and CMS and CLIA are supportive of this. We're going to augment what we're saying about this to help educate the full community and on this situation where there's a significant ratio of false positives in a population where it really matters. And there's huge disruption if there is a positive, and you want to make sure it's a true positive. Okay?
Yeah, thank you. So just one last comment on that is with all those molecular tests, unlike antigen test or antibody test, we all have those sensitivity or specificity tested and given to public. But for molecular tests, there's no such things given. I think it's hard to determine the specificity as we just discussed. And because some physicians ask me, what's the specificity of your test? Well, if I look at the EUA, and they all say, well, this is highly specific or 100% specificity. It's really, but I know it's not. So is there any effort actually putting some, at least the specificity for some of the sensitivity for our molecular tests, some of those well-known EUA?
Yeah. So, even in the early days when positive patient samples were not readily available, and we allowed a virus to be diluted into negative patient matrix. There was still negative patient matrix allowed, and our bar, which is an EUA bar, it's not a typical FDA bar for a test. During a pandemic, in this pandemic, we allow as few as 30 independent negative patient samples to be used in the validation. In the IFU, it's listed as negative % agreement. That's for technical reasons, because you don't have a true comparator. We call it negative % agreement. So if you look at negative % agreement from the very beginning, to my knowledge, all the tests authorized have a specificity determination. It, however, may be on as few as 30 negative samples, in which case the confidence intervals around that result are quite large. And then we do provide the confidence intervals in the instructions for use with the test, so you can look at that confidence interval as well. And that's another thing that's perhaps not well understood is, okay, we're measuring the performance of these tests for this EUA. And the confidence interval is dependent in large part, it's not wholly, I'd have to think through this, on the number of samples that are tested. And if only 30 are tested, and you may measure 100% specificity or 100% NPA, but statistics tells us that it could be lower than that, and we provide the 95% confidence interval, so that you can say, oh, 95% confidence interval means you can fall as low as X% on the specificity based on the small sample number that you've tested in the validation study.
Okay, thank you so much. Thank you.
You're welcome.
And I would now like to turn the call back to Irene Ihir, as there are no additional questions at this time.
Thank you. This is Irene Ihir. We appreciate your participation and thoughtful questions. Today's presentation and transcripts will be made available on the CDRH Learn webpage at www.fda.gov/training/CDRH Learn by Thursday, October 29th. If you have additional questions about today's presentation, please email CDRH-EUA-templates@fda.hhs.gov.